After a year or more in the feeding grounds of the North Atlantic, adult salmon head back to the rivers of their origin. More dramatic than their giant leaps is their ability to tolerate the changes in salinity that occur between the sea and fresh water. The transition between the two environments is made more than once during the course of their life cycle. How do they maintain a constant composition of their body fluids in the face of such dramatic changes in external salinity? What is the key to scaling the salt barrier? It's early autumn, and in the tributaries of Scottish rivers, we can begin to trace a life history that spans both freshwater and the sea. These gravel beds are the site of a mating ritual that takes place every year. First, the female releases a stream of eggs. These are fertilized by a cloud of milt from the male. The fragile eggs can only develop in an environment of low salinity. So the early stages of the salmon's life cycle are spent in fresh water. The eggs are laid in the upper reaches of rivers where the salinity is less than 1 part per 1,000. The young alevin that hatch in early spring have enough nutrients in their yolk sac to sustain them for about a month. Once they emerge from the gravel, the fish are commonly called fry. Feeding on small aquatic organisms, they double in weight within a few weeks and are known as parr. These fish have distinct markings on their sides and remain in fresh water for up to 4 years. During this time, they reach an important transition stage, the smolt. Smolting occurs only in the spring, but the resulting smolts may be anything from 1 to 4 years old. The smolts undergo dramatic physiological and biochemical changes to equip them for migration to the sea, where the salinity may be as high as 35 parts per 1,000. The maturing adults spend up to 4 years feeding at sea before returning to their native rivers to spawn. So transitions between freshwater and seawater occur at least twice during the life history of the salmon, once as young smolts migrating downstream to the sea, and a second time as adults returning to the rivers to spawn. The fish that survive after spawning may cross this salinity barrier yet again if they return to the sea and begin another spawning cycle. The River Dee and its tributaries are the site of a long-term research project on the movements and behaviour of Atlantic salmon. By monitoring different stages in the salmon's life cycle, scientists at the Marine Laboratory in Aberdeen have provided an insight into the physiological adaptations that accompany the transition from freshwater to the sea. An important focus for these studies are the smolts. The trapping of these fish is most successful in early spring when they begin their downstream journey to the sea. This migration is thought to be initiated by changing day length, and hormones such as thyroxine and cortisol are believed to be important in regulating the development of the smolts. But how can we follow the fate of these fish as they make the transition from freshwater to the sea? To answer this, special methods of tagging are used. An X-ray readable tag is inserted into the snout of the fish and checked with a metal detector. so the movements of individual fish can be followed as they migrate from fresh to salt water. What sorts of physiological problems would salmon encounter in these two different environments? Let's start with the parr, which is adapted to a freshwater existence. The total concentration of salts in the body fluids, as measured by osmolarity in milliosmoles per litre, is about 350 times greater than in the surrounding fresh water. This reflects the higher concentration of inorganic salts such as sodium and chloride within the plasma. The difference in osmolarity between the fish and its surroundings has two main effects. The fish will tend to gain water by osmosis, rather like a sponge. It will also tend to lose salts by diffusion across any permeable surface such as the skin or the gills. For stages that are adapted to seawater, such as the maturing adult, the problem is reversed. Here, the osmolarity of the surrounding seawater is about 1,000 milliosmoles per litre, which is 3 times more concentrated than in the salmon's body fluids. The fish will now tend to lose water by osmosis and gain salts by diffusion. To explore how the fish adapt to these dramatic changes in salt concentration, a series of transfer experiments is being carried out at the Freshwater Fisheries Laboratory in Pitlochry. These smolts are being removed from the freshwater tank in which they've been maintained for the past few months. They will be transferred into bins containing salt water maintained at a salinity of 32 parts per thousand, similar to seawater. Such transfer experiments can be used to simulate the transitions from fresh to salt water that occur naturally in the salmon's life cycle. The actual rate at which such transitions occur in the wild will of course vary with how steeply rivers fall down to the sea. But what osmotic stresses will the smolts encounter during the transfer? Remember that when smolts move into seawater, they become exposed to a medium where the osmolarity is about 1,000 milliosmoles per litre, 3 times higher than in their body fluids. They will tend to lose water by osmosis and gain salts by diffusion. In order to maintain their body fluids at a constant level, they need mechanisms for replacing the lost water and excreting the excess salts. We can follow the net movement of water after the transfer by looking at variations in the salmon's rate of drinking and their urine flow. Results show that the drinking rate rises about 7-fold within 100 hours after transfer, about 4 days. The rate of urine flow drops 5-fold within 30 hours. So the salmon compensates for the osmotic loss of water across its body surface with a high drinking rate and a low urine flow. But how is salt balance achieved during the adaptation to seawater? The major ions in body fluids, such as the plasma, are sodium and chloride. We can follow their net movement during the adaptation to seawater by analysing blood samples from fish removed at different times after the transfer. The concentration of sodium ions is measured by flame photometry. When a diluted plasma sample is drawn through the flame, sodium atoms will emit radiation in the visible spectrum. Under the appropriate conditions, the intensity of the emission gives a direct reading of the sodium ion concentration in the sample. The concentration of chloride ions is measured by their interaction with silver ions in solution. Techniques like this can't be used to follow the actual fluxus of ions into or out of the fish during its adaptation to seawater. But they can give a measure of the final salt concentrations that result from such movements. so they provide an estimate of the net salt uptake or loss during the adaptation. What results are obtained from the smolt transfer? Starting with the plasma sodium level, we can see that this first rises sharply on transfer into seawater. But after about 70 hours, or 3 days, the level falls to a final value that's just above the original freshwater reading. Similar results are obtained for chloride. What lies behind these results? The initial rise in blood salt concentration reflects the passive influx of salts into the blood from the surrounding seawater. But within 3 days, mechanisms are activated to reduce the effects of this influx. Firstly, significant amounts of sodium and chloride are excreted by the gills into the surrounding medium. This occurs against a concentration gradient and so must involve active transport. At the same time, we've seen how the fish drinks seawater to counteract the water loss. Since seawater is more concentrated than the plasma, this presents a potential problem by causing the concentration of the salts in the blood to rise. To counteract this, sodium in the gut is reabsorbed into the blood and then excreted out via the gills. Any magnesium ions that are taken in are excreted by the kidney. The final salt concentration of the urine is similar to that of the blood plasma. So adaptations in both kidney and gill function are important in maintaining a constant composition of body fluids, despite the effects of osmosis and diffusion. Recent studies have emphasized the role of the gills in the adaptation. If we look in more detail at the structure of the gills, we see that they basically consist of a gill arch that supports fine filaments called primary lamellae. These in turn bear finer filaments called secondary lamellae, which are the sites for gas exchange. It turns out that in between these secondary lamellae, concentrated at their base, are clusters of special cells called chloride cells that are responsible for the active transport of both sodium and chloride ions. Electron micrographs show that the chloride cells are rich in mitochondria, which are important in all energy-requiring processes. They also contain a dense tubular network of membranes that contain the ion pumping sites. In smolts adapted to seawater, this tubular network becomes more highly developed. The chloride cells are larger and more numerous. They also become associated with special accessory cells that are believed to cooperate in the act of transport. It's these modifications in gill structure that enable the young salmon to tolerate the switch from fresh to seawater. In fact, transfer experiments have been carried out to monitor the survival rates of juvenile salmon as they develop into smolts. In February, at the start of the smolting season, all the fish die within 24 hours of transfer into seawater. But by May, towards the end of the smolting period, 80% of the fish survive a full 5 days after the transfer. It appears that even in fresh water, the young polp begins to develop most of the modifications in gill structure that are eventually observed in saltwater adapted smolts. The transfer to seawater acts only as a final stimulus to bring about subtle adjustments between the fish and its new environment. The period at sea may last up to 4 years. Many of the smolts initially tagged in Scottish rivers may be captured as far afield as Greenland. Some are lost to predators. The small proportion that do return can be intercepted at the river estuaries before they make the transition back into fresh water. It's early autumn, and at Aberdeen Harbour, the netting of these returning adults has begun. The timing of entry into the estuaries may depend on a variety of factors, including temperature and salt gradients between the sea and fresh water. Although the fish may return at any time of the year, most that are caught now will be like this one, a grilse that has spent one winter away at sea.
That's a good one, Alistair.
After being anaesthetised, the fish is tagged. This allows scientists to track the rate at which the fish move from seawater to freshwater.
65 centimetres.
65 centimetre, and it's a grilse.
A grilse. And the fly tag will be 24486. Okay, just double check. 24485.
A radio tag is inserted into the stomach of the fish, with standard tags lasting about 3 months and long life ones up to 8.
Okay,
I think that's good.
fine. Now some scales.
A sample of scales gives a measure of the age of the fish at the time of capture.
Okay? Okay. Right. That's fine. Once he's recovered, Dodd, we'll go out into mid-channel and let me go. That's him.
Once the fish have entered the lower reaches of the river, tracking can begin in earnest. The signals are more easily detected when the fish is in freshwater, since the transmission of the radio waves is more efficient here than in the higher saline environment of the river estuaries. A handheld antenna picks up the specific frequencies emitted by the radio tags. By scanning through the available range of frequencies and rotating the antenna to the point of a maximum signal, it's possible to locate the position of the fish.
It's just down here in the long grass. I've put the aerial up this tree so it gets good coverage right down the river.
Oh, it's a good place.
If the salmon are moving rapidly, automatic receiving stations are used. Each receiver scans through 10 channels with preset frequencies at regular intervals.
Okay, I think they're all
This
fine.
gives a printout of the time and channel at which a signal is detected.
And we've got signal.
Oh,
There we go,
yes.
look.
The fish has gone through. What time?
9 o'clock last night on Channel 4.
Or it could be at Tilbury's by now.
That's right. It could be even further. It might be at Park.
Results show that the fish can make their way rapidly up the lower reaches of the river, sometimes travelling over 20 kilometres in a day. Does this imply an equally rapid adjustment to the freshwater conditions? The answer to this is also being explored at the Pitlochry laboratories. Adult salmon, fresh run from the sea, are used in a separate set of transfer experiments. The fish are first removed from seawater and anaesthetised. They are transferred to an experimental seawater tank once a catheter has been inserted into the urinary bladder. This allows urine samples to be collected at intervals during the experiment. After allowing for a period of recovery, seawater in the tank is replaced by flowing fresh water. The changeover of the two media can be monitored by the decrease in the electrical conductivity of the water. Blood samples can be taken at regular intervals through a cannula attached to the dorsal aorta. These can be analysed for sodium and chloride concentrations as before. What osmotic stresses are posed by the transfer experiments? We know that adult salmon moving into freshwater are faced with an influx of water by osmosis and a loss of salts by diffusion. How did they get rid of the excess water? Two main changes are observed. First of all, the adult fish stop drinking on transfer from seawater to freshwater. Secondly, their rate of urine flow rises about fourfold within a period of 60 hours, 2.5 days. This high rate of urine flow indicates that a large amount of plasma is being filtered by the kidney. Salts are in fact reabsorbed from this primary filtrate so that the final concentration is about 1/5 of that in the plasma. That is, it's a dilute urine. How rapidly do the plasma levels of sodium and chloride change during the adaptation? The concentration of sodium ions in salmon adapted to seawater falls sharply within the first 24 hours of transfer into fresh water. But after that, it begins to level off so that after 60 hours exposure, the sodium levels have stabilized to a value somewhat below that in seawater. A similar pattern emerges for the chloride ions. How can we explain these results? Remember that the concentration of dissolved salts in the blood is about 350 times greater than in the surrounding fresh water. The initial drop in the plasma salt concentration reflects the passive movement of salts from the blood into the external medium down their concentration gradient. However, after a day, mechanisms are apparently switched on to reduce this passive escape of salts. This time, significant amounts of sodium and chloride are actively taken up by the gills. The chloride cells are again important in the movement of these ions against their concentration gradient. So the osmotic stresses posed by movement between the sea and fresh water are met by adaptations in the kidney and the gills. It's a problem faced not only by other migratory species like the eel, but by fish that inhabit the intertidal zone, where wide variations in salinity can occur within a day. The results from salmon show that by combining field studies with laboratory analysis, it's now possible to follow both the scale and timing of these remarkable adaptations.
Included